Hi, my name is Alexis Marshall and I'm a research fellow at the University of Waikato in New Zealand as part of the Thermophile Research Group. And one of the first things that you'll want to do when you receive your next generation sequencing data is to have a look at it. And so today I'm going to, as part of the Bioinformatics Virtual Coordination Network, I'm going to take you through a short tutorial on how to do that. When you launch the Metagenomics topic My Binder platform, this is what you'll see. You'll have a left-hand panel that has two folders, data and lesson 1, and also a few files in here. You can interact with this panel in the same way that you would on your computer. So if we were to double click data, it opens that directory up and shows you that it's full of FastQ files, compressed FastQ files. You can also go back by clicking on the folder structure above. You can interact with this in the same way that you would on your computer, in that you can rename things, you can copy, you can delete. If you were to go in here and right click on a file, you can download that file. So it works in a very similar way. Let's just go back up. The other way that you can interact with this information is from the terminal. And so if we scroll down, you'll see here that there's a terminal icon. If we double click on that, it opens up this terminal space. Now I'm aware that for many of you, this will be your first interaction with the terminal. So I'm going to take you through it quite slowly. So if we wanted to see where we were on the computer or in the terminal space, we can use the command print working directory. That tells us exactly where we are. And as you can see, it matches this part over here as well. If we wanted to see what was located within this folder, we would type the command ls, which is for list, and it tells you the names of the files and the folders that are within that directory. If we wanted a little bit more information about these files, we could type ls -l, and that would not only tell us the directories and the files that are located here, it'll also give us a date stamp, some information on the size, the location, and the ownership or executable rights to those files and folders. If we wanted to go into one of these folders, just like I did over here when I double clicked, then we can change directory. So that's CD and then data. So I want to go into the data directory. One of the shortcuts on the command line is the tab. And so if you tab out, it will try to match the words within that directory or any kind of word match you can find. And as we only have one match for DA, it just tabbed directly out the data. Press enter or return and then list L again, and we can see exactly the same information here as we would if we double clicked over here. Okay. So each of these files is appended .fastq.gz. Now gz is a compression. Many sequencing providers will compress their files before they send them to you to reduce the size. You can view a .gz file in multiple different tools. Some tools you can't and you need to uncompress them. So I'll quickly show you how to do that. Give ourselves a bit of space by returning it a couple of times. So gunzip is the command to unzip a file. We start typing in the file we're after. We tab out, type in 10 because there's multiple matches to 10. Tab out multiple matches to 56, and then we get to the read one. As you'll see, each of these samples has a read1 and a read2 file associated with it. This is very common for Illumina sequencing. You'll have in this folder, we have 1, 2, 3, 4, 5, 6 different samples. And so here we'll just uncompress the first read and then tab right out and then hit return. If we go list L again, as you can see here, this file now is uncompressed. So if we wanted to view this file, we could have viewed it as a compressed file and as a non-compressed file, because the functions to view understand the compression and read them regardless. But I just wanted to show you how to do that because some tools won't know how to uncompress a file. So if we wanted, however, to do every single file in this folder, this directory, instead of doing them one by one, which you can just by copying this command and changing the file name, we can run a loop. And so a for loop is the loop that I'm going to show you how to do right now. There's 4 different sentences for a for loop to get it to run. You start off by calling the for command, and then you set a variable. So this can be anything. This can be file, A, B, variable. It's very dependent on what makes sense to you at the time that you're running the loops. So I'll just call it for a file. Now the next command that's required is in. So it's for variable in. Now you can type out every single file name individually with a space between them, or you can use a wildcard. Now in this instance, a wildcard is the best thing to do because each of these files is appended in exactly the same way. And so I can use the asterisk as the wildcard. And so what that's saying is that I don't mind what comes before the match as long as you pick up everything that matches this particular information. And so I don't mind what comes before FastQ.gz, but I do care about the .fastq.gz part. And so I don't want it to pick up this file up here that's already been uncompressed. And so we just type in any particular word match. So you could type in .fastq.gz. You could also just type in .gz or q.gz. Any of these combinations will only identify the .gz files because that's what these files are appended in. But I tend to give myself a little bit more information just because when I come back to my codes, I like to know exactly what I was doing at that time. So this is the first file, the first line, sorry, of a for loop. So we hit return. As you can see, it's asking me for a little bit more information here. That's because the for loops have 4 lines. And so we type in for a variable that matches this particular information. I want you to do this particular command. And so in this instance, I want it to gunzip. So gunzip all of the files that match this particular word match. And so because I've thrown .fastq.gz into this variable, I then call this particular variable. And the way you do that is with $, open and close bracket. So unzip the variable, and then the last line for a for loop is done. And so as you can see, it's not asking me for any more information, but it is still flashing that it's doing something. And if you look over here, you can start to see that each of these files changes from having a .fastq.gz to just having a fastq, and then it's finished. So if we type in list L, you can see that every single file here is now unzipped. Okay. Perfect. Now, if you want to just have a look at these files, there's multiple different ways that you can do that. The way that I'm going to show you is with the command less. And so we just type in less and then box, tab out, 10, tab out, 56, tab, read 1, tab. And it opens this file up. It opens it up in small chunks. And so you can see down here that it's waiting. And so if you type return, it will start to read that file to the screen for you. But the reason I've opened this up is just to show you the format. So FastQ files come in a 4-sentence or 4-line structure. The first line gives you information about the sequencing conditions, the machine, the lane number. I'll provide a little bit more information in a link for you if you're interested in exactly what each of these mean. The second line gives you the base calls for each nucleic acid for each position. Then you have a separator, and then you have the quality metrics. And so these are Phred scores. Each of these symbols have a different score, and this is what we use when we're looking at the quality of our sequencing output. So as the machines, especially Illumina, for example, go through sequencing by synthesis, each one of these nucleic acids here, the machine gives it a quality call based on how sure it was that it is actually a C and not a G or a T or an A or an N. And so that's what each of these symbols is giving you individual base pair quality metrics for how sure the machine was that it called the right thing. And this is the information that we use when we're looking at the quality of our data and doing our trimming steps before we assemble any kind of next generation sequencing data. So if you want to quit the less, you just type Q and it'll quit that screen for you. And then, yeah, and it'll take you back to the terminal. And so you can, if we had a look at each of these files individually, you would see that there are thousands of different lines of information. And so it's very difficult to get a sense of the quality of this information without processing it slightly. And so one of the ways that you can do that very easily is using a program called FastQC. So I'm going to show you how to run that now. There's a lot of great tutorials on FastQC already available. And so I'm going to give you some links to those as well. But I'm just going to show you how to run through each of these files. So the first thing we're going to do is make a directory for all of our output to go to. And so the way you do that is with make directory space, and then we're going to call it 00 FastQC. Now, everybody has their own preferences for file structure. One of the benefits of starting off your directory structures with 00 or 01, 02, 03 is that when you come back to a data set, it allows you to very quickly work out what was done first, as opposed to steps where you repeated things and came back to them later. And so if we press return and then go list L, you can see here that I now have a new directory. Okay. Now we're going to call the FastQ software. I'm going to show you how to do that individually first, and then we're going to call it using the for loop again. And so just type in FastQC. Now, if we type -H, that'll bring up the help menu for this software or this tool, and it will tell you exactly what kind of information it's after. And so you can define an output directory, you can give it different types of file types, but it's particularly after call the program and then give me the first, second, up to the nth sequencing file that you're after. And so that's what we'll do. So if we just bring up the list of files again, FastQC, whoop, and then we'll just tab out, read one, press return. Okay. As you can see over here, it's made, when we did that, it made some new files. And so these are the two files that it's made. If we double click on the output, it will open up a new window in here. And this is what the FastQC output looks like. You have some basic statistics here It tells you what the file name was, what type of sequencing it was, how many sequences were in that file, the average length, and the GC content. It also gives you this traffic light system over here for each of the quality metrics it gives you. Now, the tutorials that I'm going to give you as a link will talk you through how to interpret this information in a little bit more detail. But what you shouldn't be too concerned about is the green, orange, and red nomenclature. And this is because depending on your sequencing chemistry or the type of data you're looking at, you will get a fail or a pass that you need to interpret based on what your data type is. As you scroll down, you'll see, if I can make it a little bit bigger, you'll see that it's graphed a lot of these quality scores. And so this is the per base sequence quality. Now, Illumina data, make this a little bit bigger. Okay. Okay, so what this graph is showing you is the first base up until the 9th, and then it's grouping bases as it goes along that sequence read. And this will change depending on the type of sequences that you've put in. But what it's showing you is those Q scores or those Fred scores on the y-axis here. And so the green is saying that, yes, the machine was very confident in its call. Orange is, well, maybe it's okay, but we're not as confident as we were up here. And then red is generally an indication that the machine wasn't very accurate at that particular base call position. Now, in Illumina sequencing, as you go across, as it starts to sequence the read, the qualities are high and then they do tail off towards the end. So this is a very common thing to see in this type of data. The other information you'll get here is per base quality sequence scores. You'll also see sequence content information, GC content, and also read length distribution. So there's a lot of information that this program provides you that the other tutorials will go into in more detail. So if we go back to our terminal space and we scroll back in again. Right, there we are, perfect. We zoom back in. What I'm going to do is remove these two files to begin with, just because I want to show you how to run a loop on these files and to push the output to this directory here. So if we just give ourselves a little bit of space, and then we look here, what you can use is the remove command. Now you need to be very careful when you use this because you don't want to delete files that you're not after deleting. But I'll show you how to use it. So in this instance, I can use the asterisk again for .html because there's only one file in here with that match. Press return. L, you see that it's missing now. One of the other shortcuts in the command line that's really useful is the up and down keys. And so I'm just going up at the moment. And you can see that I'm going through all of the previous commands that I've used. This is great because it means you don't have to type out everything. So I can just delete the HTML and then add the zip here. So if we go to list L, you can see that both of those files are now missing. Okay, so now we're going to call FastQC using the for loop. And so as before, we call for, we set a variable in, we use a wildcard. All of these are FastQ files now. And so that's going to match everything that's within this directory. Then for file in, I want it to do and call FastQC. We tell it the variable again, And then we give it the output directory. And so this is 0, 0, and then we can pad out because there's only one directory that matches 0, 0 in that location. And so FastQC, this is the variable. I want you to throw all of the output into this directory. And then we go return and done. And as you can see, it's outputting what it's doing to the terminal. You can capture this information as well. I can show you how to do that once it's finished. But at the moment, it's just running. And if you go over to your computer terminal area here and double click, you can start to see that these files are being generated. Take a little bit of time to get through all 6 samples because there's 2 files for each sample. Okay. Okay, as you can see, now it's finished. We can go list L. And as you can see, there's nothing in our current directory. So we want to change into the directory that we just made. And there you can see all of these files. Now, as before, we can open each of these up individually from this area and have a look at each sample independently. But it's also valuable to be able to compare each of your samples to each other. And so one of the ways that you can do that is using a program called MultiQC. Now, MultiQC will go through and look for log files that have been created by FastQC and other software as well. So later down the line, if you want to have a look at the outputs from multiple different trimming steps, you can use MultiQC to pull that information together. But what we're just doing now is to be able to compare the FastQC output from each of these, for each of these samples. And so we call MultiQC. And as before, I just have a look at the help menu. And you can see everything, all of these options that you can call as well. But MultiQC is designed to work it's very user-friendly. And so what we're going to do is just call MultiQC. And I just wanted to search the current directory. And so one of the shorthands for that is just a full stop. And so MultiQC will then run all of this output. It'll tell you where it's looking, what it's found, how many reports it's found, and quite a bit of other information. And so list L, you can see that there's been this new folder that MultiQC just created, and also this HTML report. So if we go over here and scroll down to our MultiQC report, double click on that, you'll get this window. Now, if you scroll down immediately, you'll see that it hasn't loaded some of the images, and this is because this program uses Java. And so what we need to do initially is to just go over here and click Trust HTML, and then that will open up all of these images that MultiQC creates. Just quickly, MultiQC will give you a sample summary, so general statistics. So it gives you all of the sample names and summarizes some of the important information that each program outputs. And then it'll also compare all of your samples to themselves for each of the quality statistics that FastQC provides. But each of these lines now is each of your 6 samples with the read 1 and read 2. As I said, another tutorial will go through how to interpret this data. But I hope that you found this helpful, and I hope to hear any feedback that you have as well. All right, thank you.